So thank you for having me. I'm really excited to talk about some of these topics, and hopefully it'll be relevant for everyone who's doing these kind of non-invasive surveys. So I'm a PhD candidate in John Paulson's lab at Duke University. And in 2015, our lab at Duke, in collaboration with Gabon's National Park Agency, initiated the Forest Elephant Movement and Behavioral Ecology Project. And since then, we've placed about 100 GPS collars on elephants across the country. And a quick thank you to Pete Morkel, who is a South African vet who actually helped us place all the collars. The work would not be possible without him, and our wonderful field teams who have helped us with these projects. And from there, we've kind of we follow the GPS collared elephants both on foot and via the satellite positioning. And the study that I'll talk to you guys about is a derivative of those studies. But I realize I'm the only African forest elephant researcher here at the moment, so I want to talk a little bit about the forest elephants first. Currently, they're listed as a subspecies of the African savanna elephant. However, there's mounting literature that shows that this is definitely not the case. There's several morphological, physiological, behavioral, evolutionary differences. A recent study actually even showed that forest elephants are more closely related to an extinct European long-toothed elephant as opposed to the savanna elephants. As you can see in the map, represented by the darker green, forest elephants are really limited to this tropical rainforest areas, with about 50% of the population maintained in Gabon, which is the Boxton area. Probably the most striking difference is size. Forest elephants, a large male forest elephant will only be about 2.5 meters, whereas savanna elephants can reach 4.5 meters. Forest elephants are also largely frugivorous, but they'll also eat leaves, bark, herbs in the area if it's available. But again, most of them are under this dense tropical forest. And a single site, they can eat over 300 species of fruits. So they're having large impacts on their environment, major seed dispersal, key ecosystem engineers, as you would expect from the savanna elephants. They also have a lot slower life history than savanna elephants do. So forest elephants don't reach maturity until 20-25 years, with females having their first offspring at about 23 years, which is almost twice as long as some of the savanna elephants, which also makes some of our repopulation estimates a lot slower. An average group size is only about 3 to 4 individuals. Personally, the largest group I've ever seen was in a coastal savanna, and it was 22. So much, much smaller, and a lot of my thesis work is actually looking at these environmental drivers. However, I'm going to be talking to you guys today specifically about dung degradation and these estimates that go into forest elephant work. Unfortunately, forest elephants have had large recent drastic declines. In 2013, they were estimated to be at about 100,000 elephants. And then our lab did a follow-up study in just a specific park in Gabon, which, as I mentioned previously, is the stronghold for elephants. And in this repeat survey, so there was one study in 2004, and then another survey in 2014, we saw a loss of about 20,000 elephants, which the population a few years ago was 100,000, 80,000. There's a current, Vulcan is currently sponsoring a recensusing of elephants in Gabon currently to try and get a better estimate of where the remaining forest elephants are. And this is what looking for forest elephants is like. So unfortunately, we can't just go through and count. These are photos that I took while using GPS collars and working with some of the best trackers in Gabon. So there are 2 elephants in these photos, I promise. But it kind of shows the situation of counting elephants to get a population estimate just isn't really possible. So you can, in the left photo, you can kind of see here to the right, they both have yellow collars. And then in the right, you can kind of see it's a large male, so you can see the trunk and the forehead. But yeah, it's why we use non-invasive, indirect methods for counting forest elephants. So jumping off of Cindy's talk, a lot of researchers now use transect surveys. So you walk through the forest, count as many dung piles as you can, create a density. And then from that dung density, you use a defecation rate and a dung production rate to figure out how many elephants there are. These are typically done, so defecation rate for African forest elephants, there's some clearings where people will set up an observation platform and just count how often they defecate as part of other studies. And the numbers are more in agreement. There's a range typically from about 16 to 21 dung piles per day produced. However, dung degradation rate is extremely variable based, as was mentioned earlier, based on termites or season. And so, one study might say that there's a defecation rate of 20 days, and another one will say 240 days. And I'll kind of get to the point is that this can make a large difference on your population estimate, which is what we found. So I'll walk you through this table, but this is from the Minkebe study that we had talked about earlier, where we saw a loss of 20,000 elephants. So the top part is the 2014 study, bottom part, 2004 study. I'm going to bring your attention to the blue box. And so these, we were unable to do our own degradation and decay studies. So we took numbers from nearby studies, one within Gabon and one in Congo, which estimated a dung decay of 45 days and 56 days. And this resulted in an estimate of 15,000 elephants or 12,000 elephants for that area. And so this 10-day difference in decay rate resulted in a 20% difference. And again, if our total population of forest elephants in existence is in the tens of thousands, a 20% difference just in our number estimates is huge. It can have major influence on management decisions, or what parks are we putting our attention on. And so, as I've mentioned, some studies do their own decay rates, but they're often retrospective, or where people will find dung and then return, every 6 to 8 weeks to see how long it takes the dung to disappear. But 6 to 8 weeks is far more than 10 days, which already had a 20% difference. So this was the inspiration to kind of move away from the site-based estimations and actually look at what are the environmental factors influencing dung decay, and can we create a model that will allow us to systematically look at this degradation regardless of site. So if we have our little dung pile and we're trying to imagine what is causing it to decay, what might speed up decay, what might decrease it, first thing that came to my mind was environmental factors, so sunshine or precipitation. So are we drying out the dung? Are we washing it away? What are the habitats? Is it in a savanna clearing area, or is it in a forested area, inundated forest? What is the canopy cover? What is the substrate? How quickly might it be washed away? Is there animal disturbance? If we have termites, dung beetles, red river hogs rummaging through the dung, that'll cause it to disappear a lot more quickly. Then there's also characteristics intrinsic to the dung piles themselves. So in our previous talk, we were talking about the S phases, but actually as an elephant defecates, it might break up more in certain spots than others, which will have an influence on how long it takes to disappear. Number of boli, the size. A small female elephant will not have the same size dung as a large male, and those take different amounts of time to decay. Similarly, what was the elephant eating? That will have a large influence. So as I mentioned, forest elephants are largely frugivorous, which will cause a much faster decomposition than, say, savanna elephant eating mostly herbs and browse. So, as I mentioned, we were tracking the GPS-collared elephants on foot, and so we worked in 3 sites in Gabon. And while we were following the elephants, when we found really fresh, within a couple of hours, we would GPS mark the dung, and we would return to it every week to 2 weeks to follow this degradation and record environmental variables associated with it. And we were able to do this with 188 dung piles, where we collected this local information, and then we also collected remote sense information. And so these are satellite images that represent different ecological or environmental variables, so precipitation, humidity. It could even be things like canopy cover, just at a broader scale from these satellite images. And from there, we used model selection to find which variables were most important for dung degradation. We created 3 different models, and I'll talk about each one and the motivation for each of the separate models. And then the important part is we validated it with a withheld subset of our data, because what's the point of a model if it doesn't actually do what you're hoping it to do? So we first start with a field data model. And so this is fine scale what is going on right above the dung. And the goal of this was that if you're walking a transect, or field teams are walking transects, they can collect this information at the same time, so we're not spending time and money just watching dung disappear. And so you have the here and now, what are the environmental factors influencing the dung decay? And so we looked at information like animal disturbance, substrate, habitat type, canopy cover, number of boli, and the seeds as an approximation of diet. And we found really that animal disturbance and the number of boli were less important, but substrate, habitat type, canopy cover, number of seeds had a large influence on decay. We then switched to our remote sensing. So this is again a more broad scale, as the finest would be about 30 meters. So we looked at slope, temperature, vegetation complexity, which is a good kind of proxy for habitat type, precipitation, broad scale canopy cover, and temperature. And what's really cool about remote sensing is that most satellite imagery is available back to 2000. So any surveys that's been done in the last 2 decades, we can actually now use one systematic method to estimate decay and now create a cohesive population estimation instead of juggling, is this the same season, is it under the same rain regime? We can have these cohesive studies. And so we found that precipitation, canopy cover, and humidity were the relevant variables. So it's kind of nice to see that both at the broad scale and the fine scale, some of these variables, such as canopy cover, are consistently important. And finally, the idea was the best of both worlds. If you can have your field teams collect the information, you can get this free satellite imagery. Why not use all the information available to us, have a better model, et cetera. And so some of the variables turned out to not be as important in this mixed model. Habitat type, humidity, tree canopy cover kind of fell out. So, have our variables, how well did they do? Actually decently. So on the left, in the blue box, is 1/3 of our data which was withheld. And the line in the middle of the box is the median, so we had about 27, 28 days time for our dung to degrade. And then the 3 white box plots next to them are the 3 different models that we made trying to estimate that same median. And we were pretty close, so we had an estimate of about 24 days for degradation, which is great because that wouldn't have too much of an influence on our population estimates. And we were graciously given a study from Congo. So the next step will be actually looking at how well does our model predict outside of the study area. Can we now use this model regionally? And I have high hopes, and we'll see where it goes. So going slowly, going back to our equation, we're still trying to guess number of forest elephants, and to kind of drive this message home of why it really matters. As I mentioned, WCS is currently redoing surveys in Gabon, and so they were recently in Avindo National Park, which is about 3,000 square kilometers, and they found an average of about 1,300 dung per kilometer squared. And so we wanted to look at, based on different precipitation and habitat variables within Avindo National Park, what could be the estimate variance that we would get depending on what season it is. And so we were just looking at if we tweaked the precipitation range, and what we found is that if you only had like 1 millimeter of rain in a dry season, you'd expect a degradation of about 36 days. If you went to the other extreme, a high rainy season with an average of about 10 millimeters of rain per day, you'd have about 24 days to degrade. And then, so in the plot, you have the amount of rain, and that's elephant estimations. We have a 30% difference. And so, again, to put this in context, what it means is that if you were to go and, regardless of the season, just see these 1,300 dung piles around, you could estimate 10,000 elephants, or you could estimate 7,000 elephants, which has a huge difference. Or if you did a previous study a few years back, and saw 1,300 dung piles, but it was in the rainy season, and 1,300 dung piles now in the dry season, we would have a loss of about 3,000 elephants, or 30% of our elephants. So this should hopefully be key in moving forward in these long-term studies to keep a consistent estimation method. So this model should hopefully be usable for any elephant species or great apes, or any animal that we're using non-invasive methods that required a degradation rate for dung or nests. It's great for forested environments where we're not observing our animals. It's great for multi-site studies where degradation on the coast might not be the same as inland, or some areas might be much more swampy. And then repeated studies over time, or studies that are spanning seasons, we can now incorporate these changes in temperature, humidity, rainfall. And it reduces a lot of this observer bias that we could have from one year to the next. So I hope this is useful for some people. We're also looking into other kind of estimate issues with woodline transects, such as deposition rates, or how often are the elephants defecating? What are our observers' abilities to accurately decipher two different defecation events, or what are the chances that we're resampling the same elephant across transects? So we're kind of working to improve these methods because it's the best that we got right now to count our elephants. A big thank you. This project was entirely funded by the International Elephant Foundation, and then FEMBE as a whole has had a lot of support, so we really appreciate it. I think we should have some time for questions.
Thank you.